process and allows various chemical reactions to occur. But these are more than just reactions, for even a mere respiration involves a lot of processes. But what makes this reaction happen at very fast pace? Enzymes, also known as biological catalysts, are proteins that are found within the cell which assist life by speeding up the rate of chemical reactions. They are very specific in the reaction which they catalyze, even the choice of reactants or substrates. In this experiment, let us try to determine the enzymes that are present in foods and how certain substances affect enzymes.
Hello everyone! For today's experiment, we will be needing an apple, pineapple tidbits, which will serve as our meat tenderizer since meat tenderizer powder is not available, then a vinegar, and of course, water. We will be also needing our measuring cups and measuring spoons, a jar or a clear glass, a can opener, a fork to mash the apple, a knife to slice and cut and peel the apple, and of course, a ladder, and a pan. For task 1 under Model 2 titled Juice Coenzyme, peel 1 apple and chop it into small pieces. Then, add 200 ml of water and place it in a cooking pan. Now cover it and set the heat to low. Let it simmer for at least 20 minutes or until the apple looks tender and soft. While on the pan, use a fork to mash and blend the chopped apples into a puree. For step 2 under task 1, measure 5 tablespoons of meat tenderizer powder and transfer it into the empty jar. However, since meat tenderizer powder is unavailable in the area, I made use of pineapple tidbits where I extracted its juice. The juice of a pineapple can serve as a meat tenderizer also, hence I made use of this substance instead. Then, add 150 ml of water in the jar and mix the solution. Then for step 3, place 3 tablespoons of a puree in the container. Eventually, add 5 teaspoons of vinegar into it. Then, pour a cup of the meat tenderizer solution and mix it thoroughly. Using a cloth filter, filter the solution. Afterwards, wait for 10 minutes.
Task 2. What keeps them from browning? First, I sliced half of the apple and prepared 10 cubes with nearly the same size. Then I labeled the lid of the containers as control, water, oil, lemon juice, and onion. I placed 2 cubes of apple in each container. I sliced the lemon and squeezed the juice into the bowl. Using a spoon, I put the different substances in the container. I also chopped some onions and added it to the sample. The control remained as it is. After that, I added 5 teaspoons of vinegar in each container. Lastly, I refrigerated it overnight.
First step, put 1/4 cup of hydrogen peroxide in 4 separate plastic cups. For the second step, put a cube of potato in cup 1, cube of onion in cup 2, cube of apple in cup 3, and cube of pineapple in cup 4. Add 5 teaspoons of vinegar in each container. Compare the 4 cubes in terms of the bubbles produced.
Hi guys, I'm glad you have arrived with the fourth task entitled Magic Bubble, and these are the materials for the experiment. For the potatoes, it was sliced beforehand. I also used a ruler to ensure that the potato strip dimensions were consistent. Now let's get started. For the first step, I prepared 4 plastic cups and labeled it from 1 to 4. Now that the cups have been labeled, it's time to go on to the next step. For the second step, we prepare the mixtures for the first and second cup. We put 5 teaspoons of vinegar in cup 1. Then, we prepare for the baking soda solution, which is 1 teaspoon in half cup of water. Then 5 teaspoons of this baking soda solution will be transferred to the cup 2. Then, we put 1 strip of potato to each cup. We soak it for 5 minutes. While we wait for the time, we proceed to the next step. We put 5 teaspoons of hydrogen peroxide in cup 3 and 4. Five minutes is up, therefore, we transfer the strip from cup 1 to cup 3. Then we add 5 teaspoons of vinegar in each container. We transfer the strip from cup 2 to cup 4. Add 5 teaspoons of vinegar in each container.
Heat first the lemon juice.
Prepare 2 glasses of 100 ml fresh milk. Add 1 teaspoon of lemon juice to the first glass. Add 1 teaspoon of heated lemon juice to the second glass. Stir the solution and record for the results in 5 minutes.
These are the observations observed in the experiment. Task 1. Slicing and cutting through the apple exposes the fruit's connective tissues to oxygen, hence causing its appearance to start to turn brown. However, when the sliced apple was submerged in water, the browning of its appearance slowed down. Comparing the filtrate as soon as it was filtered and its state after 10 minutes, there were no visible changes in its physical appearance. The filtrate remains hazy with a burnt orange to brown color since it was already fused with a milk condenser and vinegar, which both inhibits the enzymatic browning due to their high acidity. Task 2. The browning in the solution can hardly be noticed, and they have a white and light colored appearance. The liquid solution in the container turned cloudy, while the one with the onion turned pink. It is almost impossible to rank them because all of them had light appearance at first glance. The last picture shows the ranking of the samples based on browning with the oil, leaving extensive browning among the 5 but hardly be noticed, followed by onion, water, control, and lemon juice, being the lightest in color. Task 3. Out of the 4 cups, the accumulation of bubbles were prominent in the onion sample, where bubbles can be seen at the surface of the solution and surrounding the onion. For the other samples, like the potato and apple, tiny but large quantities of bubbles can be seen surrounding the cubes of the sample. Also, the pineapple cube sank at the bottom of the solution, but there were also bubbles present surrounding it, but lesser than the other samples. Task 4. In the first cup, there were no major changes before and after, as the solution only contained vinegar, while in the third cup, containing hydrogen peroxide, it was clear at first but became cloudy. Unfortunately, no changes or reactions happened in the cup. Meanwhile, when only the baking soda solution was added to the second cup, it was clear, but when the vinegar was added, the mixture released a piece of carbon dioxide that bubbled up, rising into the top of the mixture. Lastly, the fourth cup was initially clear but became a little bit hazy when the potato strip dipped into the baking soda solution from cup 2 and a 5 teaspoons of vinegar were added. The mixture did not any cause reaction other than its physical appearance. Task 5. Both the two glasses have clump formations, but the glass with the heated lemon juice has more clump formations than the glass with the non-heated lemon juice. The kernels of milk are scooped in the first glass, not the second glass. When being poured on a stainless steel basin, you can surely see the kernel formations. In terms of odor, the first glass is somewhat sour in odor, and the second still has the milk odor. In appearance, the first glass is darker in color than the second glass, which appears whiter. Our conclusion: enzyme experiments, even at home, are vital for our comprehension on how different conditions impact the rate at which enzyme reactions occur. The activities provided were easy to understand and prepare, as it compelled us to use common household equipment. Thus, we were able to meet the given objectives, including the analysis of the presence of enzymes in various sources and recognition of their roles and mechanisms. Also, by understanding the implication of the different factors influencing enzyme activity, we realize how important it is for enzymes to operate in optimum environmental conditions.